All right, so let's talk about the basics of circulation and the circulatory system. So the circulatory system consists of arteries, veins, capillaries, and the heart. We're going to talk about the heart later on. But right now, what I really want to talk about is arteries, veins, and capillaries. Arteries are defined as things which carry blood away from the heart, whether that blood is oxygenated or not. If you're carrying blood away from the heart, you're an artery, and most arteries are structured fairly similarly. The structure of arteries doesn't depend on whether they're holding oxygenated or deoxygenated blood. It depends on whether or not they're feeling the systolic pressure from the heart. Arteries have to be built the way that they are built because they get blood in huge boluses that are pumped suddenly from the heart. They have dramatic swings between systolic and diastolic pressures when the heart is beating, when the heart is not beating. That's what determines the structure of arteries. It doesn't matter whether the blood is oxygenated or not. Veins carry blood back from the heart. Capillaries connect the smallest arteries to the smallest veins and have a very thin-walled structure that allows for gas and fluid exchange to take place. You can distinguish between arteries and veins. So this that you see up here, this is a neurovascular bundle. This is fairly commonly seen where you have an artery, a nerve, and a vein all running together. And this up here, that's an artery. Arteries and veins have basically the same structures, but they're going to be of different thickness and robustness. All arteries and veins, not capillaries, but all arteries and veins have the same three tunics. The Tunica Interna, which consists primarily of endothelium, which is an epithelial layer, typically a squamous epithelial layer, a simple squamous epithelial layer. And even capillaries have that. In fact, that's kind of all that capillaries are. But moving outwards from there, you have the tunica media, which consists primarily of smooth muscle, and the tunica externa, which consists of connective tissues, dense, irregular connective tissue for the most part, as well as in some of the larger blood vessels, elastic tissue. And here you can see this is a fairly large blood vessel, and you can see this layer of elastic tissue right there. That allows the artery to stretch when it receives this big bolus of blood from the heart. In veins, you're going to have basically the same tunica interna. The tunica media, while present, will typically be much thinner. And the tunica externa is still fairly robust, but often does not include as much elastic tissue. You're less likely to find elastic tissue in veins because veins don't have to stretch as much as arteries do. So let's do a deep dive into these layers. The tunica interna, again, consists primarily of the endothelium. The endothelium is a specialized epithelial layer. The epithelium that faces the inside of blood vessels and the inside of the heart is the endothelium. It's typically simple squamous epithelial cells overlaying a basement membrane and a little bit of some loose connective tissue, primarily areolar. Endothelium is useful for a number of reasons. The endothelial lining has proteins on it that are going to repel platelets. It allows for damaged blood vessels to be healed and replenished, since endothelial tissue or epithelial tissue can be regenerated, at least if it suffers fairly minor damage. And squamous epithelial cells are very, very thin and allow for easy gas exchange. So it acts as a selectively permeable barrier. It secretes chemicals that stimulate dilation and constriction of the vessel. Some blood vessels can auto-regulate themselves based on their dilation and constriction. They're also under the control of the sympathetic, parasympathetic, mostly sympathetic nervous system, but they can control themselves based on local conditions as well. The endothelial tissues respond to inflammatory signals by producing cell adhesion molecules that basically slow leukocytes down, especially neutrophils, and then those leukocytes basically attach to the endothelial tissue and then extrude through the endothelium into the inflamed tissue on the other side. Endothelial tissues actually respond to histamine and other inflammation-inducing chemicals by changing their shape and nature. They produce these cell adhesion molecules, and they also become more permeable. Usually, blood vessels do not allow cells to leak out. It's important because you don't want blood cells to leak out. But under the influence of inflammatory signals, they become much more permeable to white blood cells. The tunica media and externa, well, the media consists of smooth muscle, collagen, and elastic tissue. It is thicker, much thicker in arteries than veins, and it is the primary thing that determines the diameter of the blood vessel. It also strengthens the blood vessel and prevents tearing, prevents blood pressure from rupturing them. The tunica externa, sometimes called the tunica adventitia as well, is primarily loose connective tissue that can actually spread out and not necessarily have a defined outer border, right? So the tunica externa of a artery may kind of meld with the tunica externa of a neighboring vein, and the epineurium of a neighboring nerve, and it may actually have much smaller blood vessels or even capillaries running through it. So this anchors the blood vessel, provides passage for small nerves, lymphatic vessels, and the tunica externa contains the vasa vasorum, because large blood vessels, if you think about it, right? Okay, you've got a large blood vessel, and it's carrying lots and lots of red blood cells inside of it, but oxygen, while it can diffuse through the epithelium fairly easily, can't diffuse through the tunica media, the smooth muscle cells, and all of the thick connective tissue, right? So you'll actually have these little branch arteries that carry blood out to that connective tissue where you can provide oxygen to the cells in the tunica externa itself. These tiny blood vessels that branch off from the artery to provide nutrients to the arteries and veins themselves are the vasa vasorum. There are several different classifications of arteries and veins depending upon their size and where they're found. The largest arteries are called conducting arteries. These are your superhighways, right? Your aorta, your femoral arteries to a certain extent, your carotid arteries. Basically the aorta and the first set of branches coming off of it. Conducting arteries are big and they have a lot of elastic tissue. And here you can see the elastic tissue. And the reason for the elastic tissue is because these are what's actually feeling the pump from the heart. When all of that blood comes rushing out of the heart, it's going to exert more pressure on the arteries, and in order not to rupture, these conducting arteries need to be able to stretch without breaking. Hence, the elastic tissue. This is actually one of the few places where you'll find abundant elastic tissue in the body. The tunica media is actually it's mostly elastic tissue, actually. It has relatively little smooth muscle because you don't really change the diameter of your aorta, right? There's no reason you would ever want to constrict your aorta. You should always have blood flowing through your aorta. Medium arteries are called distributing arteries. These are like your, kind of your main highways, like not your super freeways or something like that, but these are your state highways sort of things. They branch off of the conducting arteries. So if your conducting artery, comes out of your heart and goes down the aorta, and this is your conducting artery down the. All right, I'm probably into the arms as well. All right, so those are your conducting arteries. All of the arteries that come off of there and go to like specific places, those are distributing arteries. So like your hepatic artery that's going to take blood to your liver. Let's draw a little liver in here. Your mesentery artery that's going to take blood to your intestines. You've got your, radial and ulnar arteries. A whole set of arteries that are going to take blood down your legs to various muscles within your legs. Your splenic artery. You've got your you got your superior and inferior mesenteries, gastric artery, all of your intercostal arteries. All right. Those are all coming off of the main line. They're not gas exchange areas, but they're basically channeling the blood to all of the various places that it needs to go, all of the numerous organs. Distributing arteries have a tunica interna, much the same, endothelium, basement membrane. Thin internal elastic lamina. These need to be a little bit stretchy, but not super stretchy. They have a somewhat thicker and more muscular, at least relatively thicker, and by percentage, their tunica media is much more smooth muscle, less elastic tissue. An external elastic lamina and a tunica externa. Right? These are then going to branch into a huge number of arterioles. All right, let's actually. And these arterioles are going to come off of every artery and penetrate every tissue. You have more arterioles in your body than anything else. You have miles and miles and miles of lengths of arterioles. These things are typically so numerous and so small that they don't have like individual names. They're just called the arterioles of the intercostal or the arterioles of the brachial artery or something like that. These are going to head off into capillary beds. Arterioles, since they're really small, and because you have so much of them, these are actually where most of your resistance takes place. So when you send down vasoconstriction or vasodilation signals, for the most part, that's affecting the arterioles. It's not very much affecting your distributing arteries, and not really affecting your conducting arteries at all. Mostly it's going to the arterioles. So arterioles have a tunica interna, which is pretty much the same as it was before, basement membrane, relatively thick, at least for their size, tunica media, which is very muscular, smooth muscle, and a relatively thinner tunica externa. These arterioles have capillaries that branch off of them. And so each of these arterioles is going to branch into a set of capillaries. And capillaries are where the gas exchange is going to take place. And you're going to actually get because in capillaries, you have just an endothelium, and an endothelium is thin enough that you can get gas exchange happening. And so you see here, the capillaries are where gas exchange happens. You drop off your oxygen, and then capillaries are going to collect into venules. Which are basically the equivalent of arterioles. They're small veins. They have all three layers, an interna, media, and externa. Relatively speaking, their media is very thin. You don't control much blood pressure with your venules, and they don't do a whole lot of dilating or contracting. Venules are going to come together to make medium veins, which are what is going to return blood from various organs. And medium veins are the veins where you have lots of valves in the endothelium to make sure that the blood goes the right direction. A lot of these medium veins, some are surface, some are deep. They often run inside or in between muscles, where muscles can then, like the action of muscles can squeeze them to keep blood moving. This is called venous return. Again, you have all three layers, and again, you have relatively larger diameter than the corresponding medium arteries, but a thinner tunica media without too much muscle. All of the medium veins are going to run together, basically collecting into a few major large veins. And so these are going to be stuff like your inferior vena cava, your superior vena cava, your jugular vein, main things that are just one or two steps removed from the heart itself, and that are going to bring things back to the heart. They're thicker, they're larger even than their corresponding conducting arteries. They have a, well, relatively thicker tunica media than a medium vein, though it's still thinner than a conducting artery, and it doesn't contain a whole lot of elastic tissue. It doesn't need to stretch like that. But it does have a fair amount of smooth muscle. That just helps make it tougher so that it's less likely to rupture or split or anything like that. Arteries are where most blood resistance takes place. They're also where, like, your heart pumps blood into your arteries. The heart is the main force that forces blood into the arteries, and the arteries are the main thing that resists flow. Where you have flow resistance, it mostly happens in arteries, and so arteries are sometimes called resistance vessels. This is basically just what I talked about before. Conducting arteries are sometimes called elastic or large because of their thick layer of elastic tissue. And these will actually, like, expand and contract, not due to the action of smooth muscle, but just because they're kind of like, you can think of them as being sort of made of rubber. Distributing arteries distribute bloods to specific organs, your brachial, femoral, renal, splenic arteries, stuff like that. Thick, smooth muscle layers to the walls, 3/4 of wall thickness. The arterioles are where most of the resistance takes place. And that's because they are of such small diameter to start off with. The smaller the diameter, the more effect you get from, the more resistance effect that you get. And it's not linear, it's exponential. So it's actually really hard to control resistance with larger arteries. With smaller arteries, like arterioles, you can get dramatic changes in resistance by having small changes in the amount of blood vessel diameter. This is where your blood pressure is primarily determined. This is also where your, at least one of the places where you're going to control how much blood goes to particular organs, right? If you constrict the arterioles leading to the kidney, you have less blood flowing to the kidney. They have a thicker tunica media. That's the muscular portion in proportion to their lumen. Very little tunica externa. And you will also have what are called meta-arterioles. These branch off from arterioles and then connect into venules. So they link arterioles to capillaries, and thus from capillaries to venules. And to sort of show you here you can see this is a, all right, this is an arteriole. This is your venule. And this that's in between is your meta-arteriole. And blood branches off into a capillary bed from the meta-arteriole. And so the meta-arteriole will like constrict down, and that will force more blood into the capillary beds where gas exchange takes place. And then your deoxygenated blood gathers on the other side and goes into the venule. At the beginning of each of these capillaries coming off of the meta-arteriole, you can see that there's what's called a capillary sphincter, or a precapillary sphincter. This controls how much blood goes into the capillary bed versus just going straight through. That will be important later on. Capillaries are called exchange vessels. This is where gases are exchanged, oxygen leaves or enters, carbon dioxide leaves or enters, and also where nutrients, wastes, fluid, hormones, everything like that passes out of the blood vessel and into the interstitial fluid. They are basically just endothelium on top of a thin basal lamina, which is a basal membrane of thin connective tissue. You find them almost everywhere in the body, because pretty much everywhere in the body needs oxygen. Anywhere the oxygen is going to go has to have capillaries going to it. Where do you not find them? You don't find them in tendons, ligaments, epithelia, and the cornea and lens of the eye. This is why tendons and ligaments, and also I should say most cartilages don't heal very easily because they don't have much capillary penetration, and so can't heal very well. Epithelia has to be relatively thin. The absolute size of epithelia or thickness of epithelia is dependent upon how far away can you get from the blood vessel and still get oxygen. All right? This is why epithelia can't get that thick. There are three types of capillaries: continuous, fenestrated, and sinusoids. And I'm going to talk about each one. The vast, vast majority of capillaries in your body are what are called continuous capillaries. These are what you think of as capillaries. They are what we were just talking about, the vast majority of exchange media where you have gas exchange, nutrient exchange, stuff like that. Those are taking place in continuous capillaries. Continuous capillaries consist basically of a single squamous epithelial cell, or two, in this case, squamous epithelial cells, that is wrapped in a circle, right? So you got one or two squamous epithelial cells wrapped in a circle, and then it forms a tight junction with itself. And remember, that tight junction is relatively waterproof. The diameter of a capillary is just barely large enough that, like, red blood cells can easily fit through them. Continuous capillaries are called continuous because they form a tube that goes from your arteriole to the venule. All right? It's a continuous tube. It has what are called intercellular clefts, these little areas here. And these are very, very small areas that allow small molecules like glucose and hormones and water to be exchanged under the right circumstances, but nothing bigger than that. No proteins, not even large non-macromolecules. Continuous capillaries also have a contractile cell called a pericyte. Peri meaning around and cyte meaning cell. The pericyte wraps partially around the capillary, and it can contract and expand, containing the same contractile protein as muscle. And so this actually allows continuous capillaries to change their diameter somewhat and regulate blood flow, similar to the way arterioles do. Fenestrated capillaries are much rarer, and they are found in areas where you expect to see a lot of filtration and exchange of the capillary with the media around it. So you typically find it in the kidneys, where you're going to be filtering a lot of material out of the blood, and in the small intestines, where you're going to be absorbing a lot of material into the blood. The word fenestru means window in Latin. So fenestrated capillaries are windowed capillaries. These windows are actually little pores called fenestration pores. And so this fenestration capillary has a whole bunch of little holes in it. Those holes are actually plugged with a glycoprotein layer that keeps basically cells and large proteins from being able to fit through it. But this is kind of like a colander or cheesecloth or something like that, right? It's going to let lots of water and small molecules through very, very easily, but keep big things out. So this is going to let water and nutrients, small molecules, hormones, amino acids, anything like that get squeezed out very easily here, but larger things like cells and proteins do not. There is no pericyte here, so these do not have any way of regulating their diameter. Last, we have what are called sinusoid or discontinuous capillaries. So the other two types of capillaries were both continuous. One was called continuous, the other was a fenestrated or fenestrated continuous capillary. Basically what continuous means is that you've got like your arteriole, your venule, and your capillary goes from one to the other, all right? So it's continuous. It's got a beginning, it's got an end, and stuff can enter into the beginning and then go through to the end, like a road. Sinusoids are more like driveways or cul-de-sacs or something like that. With a sinusoid capillary, what you have is a whole bunch of like dead end capillaries that don't come from a arteriole or a venule or anything like that, and they all join together to form an arteriole. So here you can see this is the end of a capillary, all right? And it just like it dead ends there. This is where blood starts. And so you actually, sinusoid capillaries are the leakiest of them. You can see that the epithelial cells actually aren't even connected to each other. They're just kind of sitting there with no junctions in between them. And that means that relatively large things like blood cells, proteins, and stuff like that can actually move into the capillaries from this area. You find sinusoids in the liver, bone marrow, and spleen. The liver is where most of your blood proteins are made, and basically what you do is you make blood proteins in the liver, and then they seep into the sinusoid, and then from the sinusoid they get into the bloodstream. In the bone marrow, that's where you're making blood cells. So you make your blood cells out here, they get into the sinusoid, and then from there they join into an arteriole. So these are mostly going to be found where blood is born, either the plasma proteins or the cells, but this is your driveway, like that's where you start, and then you leave from that to get into your body's highway system. Those are sinusoid capillaries. At any one time, most of your body's blood, or at least much of your body's blood, is in the capillaries. All of your blood that's actually doing much exchange, being blood, is found in the capillaries. And we've talked before about how, like, say your body wants to get rid of some heat, right? What are you going to do? You're going to shunt blood to the capillaries in your skin, where blood will be released, blood carries heat, and heat will be released through your skin and into the world around you, and that's kind of how you that's one way that you get rid of heat. When you are, we said, undergoing a fight or flight response, a sympathetic response, you're going to divert blood away from the intestines, right? How does that happen? How do you, when we say divert blood towards or away something, what does that mean? And basically what that means is that you can shut down a capillary bed. There are these precapillary sphincters here, and if you want blood to not go to your intestines, there's two ways you can do that. You can constrict the arteriole so that there's just less blood flowing to the area, and you can also constrict these precapillary sphincters so that less or no blood gets into the capillary system. It just flows straight through and gets back into the vein. If you want more blood to go to a capillary bed system, like let's say your face flushes with heat because you've been exercising, you're going to dilate these precapillary sphincters, and that will let more blood out into there for more gas exchange. At any point in time in your body, at least when you are at rest, most of your blood, about 70% of your blood, is in the systemic circuit, meaning that's the circuit that goes from your heart to your body and then back to your heart. About 18% of your blood is in the pulmonary circuit, so that's what goes from your heart to the lungs and then back to your heart. And about 12% is in the heart, depending upon what part of the cycle the heart is in at any one time. Right? Of the blood in the systemic circuit, most of it is in veins. Remember, your veins are usually larger in diameter than your arteries, and there's less force squeezing it out of your veins. So it tends to pool in your veins, particularly when you are at rest. About 5% of your blood is in your capillaries at any one time, and about 11% in your arteries. Blood in the veins is subject to relatively low blood pressure, at least under normal circumstances. Veins have thinner walls that are flaccid, less muscular, and with less elastic tissue. So we already generally talked about the classifications of veins. You have post-capillary venules. These are the smallest veins. They're fairly porous. So this is actually kind of like fenestrated capillaries. So fluid can exchange in the post-capillary venules. This is actually the main place where fluid, water specifically, is exchanged with the surrounding area. So if you have blood that pools in the post-capillary venules, pressure builds up there, more water gets squeezed out, and you end up suffering from edema. Leukocytes, since this is a porous area, leukocytes can immigrate from the bloodstream through the venule walls. You have muscular venules, which are after the post-capillary venules. They have 1 or 2 layers of smooth muscle in the tunica media. That's not much, right? But they've got at least some muscle in the tunica media. Post-capillary venules basically don't have a tunica media, or their tunica media consists just of a small elastic area of connective tissue. Then you go to medium veins, up to 10 millimeters in diameter, with a thin tunica media, so they have some muscle, but not a whole ton, and a thick tunica externa, right? Because they're going to be what's holding most of the blood, so they got to have a lot of that connective tissue around it. These medium veins are where you're going to have venous valves. Breakdown of those valves can result in varicose veins, pooling of blood in the veins of the lower extremities. These are also the veins that are going to take advantage of the skeletal muscle pump to propel venous blood back towards the heart. Very large, not even really veins so much as like venous chambers, like big venous areas where you get like a lot of veins that kind of pool into, are called venous sinuses. They have especially thin walls, very large lumens, and no smooth muscle. The dural venous sinus and the coronary sinus of the heart are examples of these. There's no vasomotor response there. Blood just sort of pools in there, and they provide a repository of blood that can be tapped. Large veins, this is mostly going to be your superior and inferior vena cava, pulmonary veins, internal jugular veins, and renal veins are large veins. They have a thin tunica media with a moderate amount of smooth muscle, thick tunica externa, containing longitudinal bundles of smooth muscle, so they can actually kind of like shorten and lengthen themselves if they have to. Last subject is circulatory routes. The most common circulatory route, the one that we think of most of the time, is blood comes from the heart, goes out to the system or the lungs, where it hits a capillary bed, exchanges gases, pools back into veins, and then goes back to the heart. Your pulmonary circuit is a simple pathway. Most of your systemic circuit is a simple pathway. You go from the heart, through arteries, capillary bed, veins, back to heart. There are a few other possibilities. One is a portal system. This is found in endocrine organs, as well as in a few places where you have something that's going to be done to the blood, for instance, in the liver. All right? So in this case, let's take the liver, right? So you have your heart pumps blood into arteries. That blood goes from the arteries into a capillary bed where they drop off their oxygen. It collects back into a vein. That vein then goes to, say, the liver or some other organ, where it branches out into a second capillary bed. The purpose of the second capillary bed is not to exchange oxygen, you've already dropped off your oxygen, but often to allow for the exchange of something else, either hormones, as takes place in the hypophyseal portal system, that's in your pituitary, or to basically cleanse and filter the blood, as happens in the liver, and then the capillaries connect on the other side back into a vein that then goes back to the heart, right? That's called a portal system, and your second capillary bed is called is the portal system, and the vein that connects the two capillaries beds together is called a portal vein. That's a portal vein right there. All right, next you can have an anastomosis. An anastomosis connects an artery directly to a vein, or I should say, an anastomosis connects a blood vessel directly to another blood vessel without going through a capillary bed intermediate. You can connect an artery directly to a vein bypassing the capillary bed. This is called an arteriovenous anastomosis. They're actually less common. You can also have an anastomosis that shuffles blood between one artery and a different artery. That's an arterial anastomosis. So you could go from the heart to your arteries, right, to a small artery or an arteriole, and then get onto a different arteriole, and then branch off to the capillary bed there. You can also have venous anastomoses, where you can move blood directly from one vein to another vein without going through a capillary intermediate. That's a venous anastomosis. All right, so in summary, all right, you need to know pulmonary versus systemic circulation. You need to know the types of arteries and veins. And that's like conducting artery, resistance artery, large vein, medium vein, arteriole, venule, that sort of thing. You need to know the three types of capillaries and what their distinguishing features are and where you would find them. You need to know what a portal system is, and at least one example of where you would find a portal system of the body, and you need to be able to recognize a portal system when one is described to you. You should also know what anastomoses are and the three types of anastomosis. You need to know how capillaries, capillary beds, work. And rough percentages of where blood is in the system. So that's like most of the blood in the systemic circuit is going to be found in the veins, right? Next, arteries, then capillaries. More blood is going to be found in the systemic circulation, then a pulmonary circulation, and then the heart, that sort of thing. All right, and I think that should provide a fairly good foundation.